A very good evening. On behalf of Vijaya Diagnostic Center, I extend a very warm welcome to our esteemed doctors to the VDC Educational Webinar Series. Today's topic of discussion, fungal and anaerobic diagnostic modalities, both of which are clinically and diagnostically challenging. We have seen of late there have been a surge in the fungal infections, especially witnessed during the COVID pandemic. And another important entity of infectious disease are the anaerobic infections, which are often difficult to diagnose because of their fastidious growth requirements. To highlight the various diagnostic modalities available for diagnosis of these two important entities of infectious diseases, I have the privilege to introduce our speaker for today, Dr. Riya Sarkar. She is an alumnus of Kolkata Medical College and she has done her MD and DNB in microbiology. She has worked extensively on diagnosis of fungal infections. So I now hand over the proceedings to Dr. Riya.
Thank you, ma'am, for this lovely introduction. So, a very good afternoon, everyone. Let's start with the discussion on these two very important and interesting topics. I start with the fungal culture methods. Now, fungus, as we call them, the emerging threat, is one of the leading cause of a nosocomial and healthcare associated infections. It is said that the fungal pathogens rank among the top 10 of most frequently isolated pathogens in febrile patients with immunocompromised status. Fungus causes infections starting from superficial skin infections to subcutaneous and deep infections and often causing septicemia and systemic infections. The clinical picture of a fungal infection varies and can range from a tiny colonization to an active invasive fatal infection, hence difficult to diagnose and difficult to treat. In a bedside, always suspect a fungus. If you see your patient is having fever, not responding to antibiotics, and has a history of prolonged hospitalization. If the patient is neutropenic, has a history of malignancy or a solid organ transplant, again think of a fungal infection. A good laboratory diagnosis depends completely on selection of an appropriate clinical sample and its collection. Then comes the rapid transport of the sample to the laboratory as soon as possible. In the lab, we store the samples generally at 2 to 8 degrees Celsius, except for the dermatological samples that are not to be refrigerated. This storage of samples is also a vital information. Apart from this, identification of a true pathogen and ruling out the contaminants is the basic funda of a good laboratory diagnosis. These are the common samples that we get in our lab for fungal infection IDs. Similarly, in cases of hair pluckings, you should always remember that the tip of hair is not the part which we are looking for. A hair should be plucked with an entire hair bulb because that is the area where the maximum fungal pathogen colonize. For a nail sample also, we prefer taking the deep site infected area and not the superficial portion. I would like to make a special mention on corneal scraping collection. Whenever you do a corneal scraping collection, try to place it on a glass slide and send us to our laboratory in a moist. correlated clinically as well as with fungal culture. The procedure is very simple. We dissolve the sample in KOH for 1 hour in case of nail and 30 minutes in case of other samples. Then we take a drop of it under a cover slip and examine under 10x followed by 40x. I can show you the picture here. This shows thin hyaline hyphae-like structure under a KOH mount. So this we reported as fungal elements positive. Similarly is this picture. After this, we come to culture media for fungal ID. As you all must be aware of the media SDA, Sabirots dextrose agar. This is a special media for fungal culture because it provides certain environmental conditions for fungus, like the presence of peptone and dextrose, the pH towards the acidic side, and presence of antibiotics like gentamicin, chloramphenicol, and tetracycline, which inhibits the growth of bacteria, if any, present in the clinical sample. For isolation of dermatophytes, we have two specific medias, dermatophyte test medium DTN and SDA with actidione or cycloheximide. The function of this actidione being to inhibit the growth of filamentous fungi and propagate the growth of dermatophytes. After the inoculation, the tubes are incubated at 37 degrees Celsius and then we have a protocol to examine these tubes. For CSF, we examine these tubes daily in the first week. For other samples, the protocol is to examine the tube twice a week. This twice weekly continues for till 4th week and a final report of no growth is only to be given after completion of 4th week. But in the meantime, if we get any kind of growth, we see whether it's a yeast or it is a mold. If it is a yeast, we perform a gram stain and subsequently we perform the identification of the yeast with the help of MALDI and Vitek. If it is a mold, you can see in the right side, I have a picture of lactophenol cotton blue mount. We perform this for a mold. We see the position of the conidia, the morphology of the hyphae, and final diagnosis of a speciation can be given on basis of this. Now, I would like to talk about certain fungal pathogens which we have diagnosed in our laboratory. I would like to give the history as much as possible. Let's start with the case of Candidemia. Such cases often come from patients who have fever, hospitalized, neutropenic, and not responding to antibiotics. Sample types most commonly are blood. So blood is always sent in this Bactic myco-F, that is fungal culture specific Bactic media. This culture bottle is loaded in this right-sided machine which you can see, Bactic 9050, and we wait for a positive signal to arrive. Once the positive signal is there, we perform a direct gram stain from the blood to see the presence of any budding yeast cells and subsequently subculture it on blood agar and meconchi agar. In the left side, I can show you the picture of creamy pasty colonies of Candida in a blood agar. Similarly, in the middle, you can see the creamy pasty colonies in the SDA. This is a classical picture of Candida morphologically. In the right hand side, I can show you the gram stain of gram-positive budding yeast cells which was performed from this culture. This is again a classical picture of Candida. Now, once a Candida is detected, my next work is to see whether this is Candida albicans or non-albicans. For this, we have a very simple test known as a germ tube test. What do? We take 2 ml of serum, inoculate a pinch of the yeast colonies in the serum and incubate it for 2 hours. After 2 hours, I take up a drop of the serum and put it under cover slip and observe under microscope. In the left picture, if you observe keenly, you can see a elongated tube-like structure coming out from the budding yeast cell. This structure is called as a germ tube, and this slide is a germ tube positive slide. Who is germ tube positive? Only Candida albicans and Candida dubliniensis are germ tube positives. In the right side, on the other hand, we can see only the budding yeast cells and there is no elongation of the tube. Hence, it is germ tube negative, suggestive of Candida non-albicans. Now, why there is need to know whether it's albicans or non-albicans? It is because we have seen that Candida non-albicans is much more drug resistant than Candida albicans. Hence, with this small test, you get a basic idea of what antifungals you are supposed to use in your patient and how challenging the treatment might be. In the right side, we have of the Candida species, and then we perform the yeast susceptibility testing by Vitek2 machine that we have by using the YST08 card. I have provided a picture of our laboratory reporting Candida albicans. As you can see, we have a panel of 6 antifungal drugs consisting of fluconazole, micafungin, voriconazole, caspofungin, amphotericin B, and flucytosine. So this is how we report a Candida in our lab. Now, a case of Tinea corporis that recently had come to us. The patient had sent us the scaly skin scrapings from concentric lesions from his body. On STA, this yellow colored tube, we found the growth of white cottony fluffy colonies. This right tube, the red color, is the DTM tube. Normally, a DTM tube is yellow in color, but as soon as it gets exposed to a dermatophyte, it changes its color to red, hence promptly giving an indication that there is a dermatophyte present in the clinical sample. In the right side, you can see a LPCB bound where I can show you this arrow-like structure, this coiled structure. This is known as a spiral hyphae, which is classically suggestive of trichophyton mentagrophytes. I can show you another case of trichophyton mentagrophytes causing Tinea corporis, where you can see abundant fungal elements present under KOH, and also in the LPCB, we can see cluster of microconidia borne laterally on the hyphae, along with the presence of intercalary chlamydospores, again classically suggesting trichophyton mentagrophytes. This patient had given us his tiny white scales from the cheek of a child. When we performed a KOH mount on it, we saw a classical picture. You can see these grape-like clusters, or we can call it meatballs. This hyphae, they look like spaghetti, isn't it? So this is a classical case of spaghetti and meatball appearance in a Pityriasis versicolor caused by Malassezia farfar. Tinea capitis was diagnosed by us when the scaly lesions over the scalps were provided to us, and we found Microsporum species under the LPCB. Recently, we received an oral swab from a 24-year male that had come to us for routine culture. On blood agar, we found 2 colonies the next day. First is the white creamy colonies, as I told you earlier, and second, a yellowish white cottony colonies, which is better depicted in this SDA. These creamy colonies on gram stain were gram-positive budding yeast cells. We performed a germ tube on them, and it came out to be germ tube positive, hence Candida albicans. This cottony colonies, on the other hand, we performed a LPCB. We found a classical picture of Candida, a classical picture of Aspergillosis, showing sparsely septate hyphae, dichotomous branching, and on keen observation, the conidiophore was covering more than 2/3 of the vesicle, suggestive of Aspergillus flavors. Hence, we reported this as a dual infection of Candida albicans and Aspergillus flavors. A post-COVID hospitalized diabetic patient had given us a sample. Under KOH, we found broad aseptate ribbon-like hyphae, highly suggestive of mucor. Even the SDA showed overnight growth of cottony grey colonies, again suggestive of mucor species. When we performed the LPCB, the classical mucor was seen with aseptate, broad ribbon-like hyphae and a sporangium. We correlated this result with the histopathological and then examined under high power microscope. There the speciation, the hyphal arrangement and the conidial arrangements are easy to visualize, and hence the diagnosis of the species becomes easy. Now coming to a case of cryptococcosis, usually such samples are CSF what we receive from HIV positive patients. In SDA, we generally get mucoid colonies. On gram stain, gram positive yeast cells are seen, and here we do not perform an LPCB, rather we perform a specific stain for Cryptococcus, that is a negative stain, India ink. What do we see in our India ink? as you can see, a clear halo around the yeast cells. Now, why is this halo there? Because Cryptococcus is a capsulated organism. The capsule does not take up the stain and hence this halo. In our laboratory, we have the other confirmation test for Cryptococcus also. That is the cryptococcal antigen test by latex agglutination method. Few other fungal species that we have identified in our laboratory are Aspergillus fumigatus, Rhizopus species, and from two corneal scrapings, we have also grown Fusaria and Curvularia lunata. For fungal sterility check and quality check, we maintain ATCC strains of Candida, Trichophyton, and Aspergillus in our lab. Time to time, we maintain the sterility check and quality check of the media, of all the media that we have, the plain SDA, SDA with Actidion, and DTN. For MALDI and Vitek 2 cards, we also have a panel of the ATCC Candida strains, and we are enrolled in PGII-NER EQUAS program at that will be very difficult to treat. Also, in certain cases, we need to know the particular species of a fungus. Why? Because there are certain species of fungus which are intrinsically resistant to certain antifungal drugs. Hence, whenever in a minimum confusion or you expect your patient to have a fungal infection, definitely do not hesitate and send that sample to us for a fungal culture and get the accurate diagnosis. To summarize, fungal culture is best diagnosed at bedside. Select the appropriate sample, collect it properly and transport. Use QH mount as a screening method and confirm the positive QH mount with fungal culture. In case of blood cultures, always collect it aseptically. Put it in a mycological specific bactic bottle. The positive signal once we get, we perform the gram stain, subculture, and then subsequently the identification of the species and the YST pattern. It is always advisable that you follow this yeast susceptibility knows that whether this anaerobe is a pathogen or a commensal flora. But nevertheless, if untreated, these cases can become life-threatening. There are certain conditions that predispose to anaerobic associated infections. Say a small tooth extraction, a GIT surgery, a genital tract injury, human animal wounds, or introduction of soil in a wound. All this can lead to anaerobic infections. Anaerobic infections vary from head to toe, starting from brain abscess, cellulitis, dental infection, appendicitis, pelvic inflammatory disease, decubitus ulcer, so on and so forth. But they give certain clues which you need to follow very clearly to diagnose an anaerobic infection. Say if you have a history of a human or animal bite or a soil contamination of a wound, then think of anaerobes. If you get putrid odor or foul smelling pus in your patient, think of anaerobes. Again, blackening or darkening or a discoloration of the skin or pus or presence of gas in a tissue is a very big red alert sign for anaerobes. Never neglect any of these small signs before it gets too late. Now, sample collection and transport are the key important steps for a proper anaerobe to get diagnosed. A sample for culture is best when collected in a syringe and sent in a syringe. Second, anaerobic transport media should always be used for sending this particular sample. I repeat, anaerobic transport media is a mandatory thing. We never accept any sample for anaerobic culture if it is not sent in anaerobic transport media. The transportation should be as early as possible in room temperature And once it reaches the lab, the inoculation should also be as early as possible, maximum storage time being 24 hours at room temperature. This is the transport media that we use in our lab, the thioglycolate broth. All the specimens coming to us for anaerobic culture come in this media. There are certain samples that are highly unacceptable for anaerobic culture, like the bronchoalveolar lavage washings, ET aspirates, seminal fluids, urethral and vaginal secretions, rectal swab, perianal swabs. Remember, swabs are never accepted for anaerobic infections. Also, sampling from a superficial tissue is highly discouraged. Only a tissue biopsy to be collected or the sample to be collected from the deep site of the infection, collected in a syringe, put in an anaerobic transport media and then only sent to the laboratory. If you suspect an anaerobic septicemia, always send the blood in a Bactec anaerobic blood culture bottle only. In our lab, we get two types of samples, as I told you, blood and others. The blood is sent in this anaerobic blood culture bottle. This bottle has a certain mechanism. It has a specific sensor located at the base of the bottle. This sensor responds to the increased concentration of carbon dioxide produced by the metabolism of organism if present in the blood. It gives us a positive signal as soon as the sensor detects it. Once we get this positive signal, immediately we transfer this blood and inoculate it in this right-sided media. This media is known as anaerobic blood agar plate. Apart from blood, the other samples are directly inoculated in this anaerobic blood agar plate. Now, once inoculated, my next step is to maintain anaerobiasis. How do I do it? Years back, we used very cumbersome, tedious processes to maintain the anaerobiasis, like anaerobic jar, Macintosh Filldays jar, gas pack system, enoxamote, and whatnot. But they were very hazardous and very much space occupying. So we have come a long way where now we use very self-generating, very handy anaerobic bag or anaerobic pouch, which does not require any addition of water or catalyst to maintain anaerobiasis. It comes with an arrow indicated tablet, as you can see. This tablet changes its color from time to time to indicate us that whether proper anaerobic situation is maintained or not. Now, the inoculated anaerobic blood agar media is put inside this pouch. This pouch is known as Gen bag and it is incubated for 7 days at 37 degrees Celsius. Every day we check the agar plates for any growth. If after 7 days there is no growth, we declare this report as negative. But if we get any kind of growth on the blood agar, we directly perform a gram stain, identify the organism by MALDI TOF and report the organism. Special mention for Operation Theatre swabs. We use a selective media for anaerobes, that is RCM. You must all know RCM is Robertson cooked meat media. We take the swab, we inoculate it in the RCM media and incubate it. We wait for the growth or turbidity to appear. Once there is growth or turbidity, we perform a direct gram stain. To look for what? To look for this classical picture. Here what I can see, you can see a gram-positive purple color bacilli with a terminal end spore. So this terminal spore-bearing gram-positive bacilli is a classical picture of Clostridium tetani. Once we diagnose Clostridium tetani, we immediately inform the OT and suggest for fumigation. Again, like fungal culture methods, for anaerobic culture methods also, we maintain the strains, the ATCC strains of Clostridium septicum and Pseudomonas aeruginosa in our lab. Time to time, again, we maintain the quality check and the sterility check of the medias, the ready-made anaerobic blood agar media or the lots of Gen pack. We are also enrolled with the NABL accredited laboratory for inter-laboratory comparison on a half-yearly basis as an EQUUS program. All this is again to maintain the validity and accuracy of the results that you get from us. No wonder there are a lot of limitations for anaerobic culture, the most important limitation being the limited knowledge of these infections. Very few number of labs perform these cultures. It's very time consuming, very expensive and rapid methods are not available. True. But no matter what, These infections are very, very scary and definitely need immediate attention. So, a short take home message from this presentation is that fungal infections and anaerobic infections both are leading causes of life threatening infections. Since the diagnostic methods and protocols are highly limited, often they tend to get neglected. But whenever in your patient you are in doubt, you cannot understand what the problem is, or there is a minute alarming signal present, please do think of these two infections. Do not hesitate to take the specimen and send us for culture, because no matter how tedious or how difficult the entire process is, these infections require immediate attention, and early diagnosis and early treatment of such patients can actually help them save their lives. With this, I would like to close my presentation. Thank you so much for your patient hearing.
Thank you so much, Dr. Riya. You have given a very crisp and a comprehensive overview of the two most important entities of infectious diseases, the fungal and anaerobic, and especially highlighting the clinical scenario where one should suspect these infections and send the appropriate sample in an appropriate manner to achieve the highest degree of yield. And as you all are aware, we are heading towards an antibiotic apocalypse. So it's no longer the time for an empirical antibiotic therapy. It's very important and imminent that one should, whenever in doubt, whenever there is a strong suspicion, to look for other causes of infection other than the routine bacterial infections. Thank you so much for such a wonderful overview of these infections.
Thank you,
I
ma'am.
now throw the house open for discussions. If anybody has any queries, we are here to take on those questions. One can type in the chat box too if they have any queries.
I don't think we have any.
We now close this session. Look forward to further webinars, such educational webinar series. Thank you so much.
Thank you.